No, I'll let you and great. It's good now? Okay. Thanks.
Thanks, Daniel. Yeah, I'm present to a little bit different audience and coming from a little bit of a different background. So I'm an evolutionary geneticist that's now turned to studying the interactions between hosts and parasites, and especially interested in interactions between the red blood cell and Plasmodium parasites that cause malaria. So malaria parasites, malaria is caused by parasites in the genus Plasmodium. They all share features of this life cycle where they're transmitted by Anopheles mosquitoes. There are some other vectors for some species, but human malaria parasites are transmitted by Anopheles mosquito vectors that deposit parasites in the bloodstream. The parasites go through a stage in the liver, and then they emerge and invade red blood cells. And they pass through cycles of invasion, replication, and bursting of red blood cells, which is what drives the symptoms of malarial disease. And then a subset of parasites can differentiate into gametocytes, which can then be picked up by another mosquito bite and then transmitted onto another human host. So these are unicellular eukaryotes. So they're quite complex. They have about 5,000 genes in their genome. On the eukaryotic tree of life, they're over here in apicomplexan group, which is quite distant from most of the other species that we hear about or that are usually studied. And malaria is still a major global health burden. So these are some figures of recent malaria incidents globally. That has been declining over the past 20 years, but still is a major health issue. So giving some numbers to this, in 2022, there were 249 million malaria cases, 94% of those in Africa and 97% due to Plasmodium falciparum, which is the most virulent of the human malaria parasites. And there were 600,000 deaths, 95% of which were in Africa and 78% of which were in children less than 5 years old. So this is still causing a lot of disease and many deaths. But as you can see in this diagram over here, this is actually just kind of the tip of the iceberg, because actually there are more cases out there. And this is part of the reason why it's so difficult to eliminate, is because many people can carry these parasites asymptomatically, and they can not be affected and transmit parasites. We also know that in addition to the last 20 years, humans have suffered from malaria for thousands of years. So this has been much revised over time and still not, still an estimate, but think approximately 50,000 years ago is when Plasmodium falciparum split from its closest relative that we know of, Plasmodium prefalciparum, which infects chimpanzees and gorillas. So made this switch into human hosts. This is kind of around or a bit after the time when Homo sapiens migrated throughout the world and spread throughout the world. And we also know that this has, another reason we think that people have suffered from malaria for a long time is because we see that evidence in our genome. We see human alleles that seem to be maintained because they have a protective effect against malaria. Okay, so as an evolutionary geneticist, this is a really interesting scenario. We have a long-term kind of host-parasite interaction that involves ourselves and these complex parasites where we have multiple human genetic adaptations. And so one of my interests is trying to study this to understand more about coevolutionary dynamics between host and parasite. And then on the flip side, we're also really interested in studying this because this experiment, this evolution has been going on so long, it can also teach us about malaria pathogenesis and what strategies have been used and successful to fight parasites over this evolutionary time. So in my lab, we do take a lot of different approaches. We do a lot of computational analysis of genomic data sets. We look at human population genomics. We also look at primates and other parasites. So there are many other species that also have malaria parasites, and we try to learn about what's going on between their parasites and red blood cells. And then we're also doing association studies and kind of functional genomics of malaria infections to learn more about these interactions. So this, as I said, this has been known for quite a while, that there are these human genetic alleles that are thought to protect against malaria. And this malaria hypothesis was proposed around 1950, that actually this selective pressure for malaria, it could explain the incidence of some genetic disorders, where these genetic disorders are at higher frequency where malaria is prevalent because they offer protection from malaria. And this was initially proposed because of the geographic overlap between where, especially Plasmodium falciparum, this virulent parasite, is found, and where these red blood cell disorders are common. So this is sickle cell allele, showing the frequency of the sickle cell allele and incidence of Plasmodium falciparum, and there's fairly good overlap. Nowadays, we can also test this more directly by genetic association studies. So there have been several GWAS, including this one that I was involved in as a postdoc, which is the largest so far for severe malaria, looking at 9,000 severe malaria cases and about 9,000 controls across 11 populations. And this is a Manhattan plot showing across the human genome evidence for association with protection from malaria. We see that sickle cell variant at hemoglobin beta is the very top hit. This is the strongest association signal in the genome, meaning that it is more common in controls than in severe malaria cases. So what is sickle cell exactly? Sickle cell allele, the sickle cell allele is a single base mutation that changes amino acid 6 in hemoglobin beta from glutamic acid to valine. And this single amino acid change causes, this is on the next, this causes hemoglobin to polymerize when it's deoxygenated, forming long polymers that result in this classic sickle cell shape. So the idea is that the individuals who are heterozygous for this allele, they are protected, this confers protection from malaria, and they do not suffer from sickle cell disease. And so when the malaria is present, there's a selective advantage for these heterozygotes, and it's able to be maintained despite a negative consequence of having sickle cell disease in homozygotes for sickle cell allele. Okay, so sickle cell disease then, this is sort of what is thought to maintain a substantial frequency of sickle cell disease throughout the world. So sickle cell disease is also a major global health burden. And it results from this single amino acid change that causes hemoglobin to polymerize under low oxygen or deoxygenated conditions. And that causes lots of different downstream negative consequences. And even today, individuals with sickle cell disease have about 20-year lower lifespan. So this is a burden in the US, this is a burden worldwide. These numbers are just to give a ballpark idea of how many people are affected by sickle cell disease. So we seem to have this, like kind of bad situation where we have malaria, we have sickle cell disease, and they're kind of caught together. We can't get rid of one without getting rid of the other. And so maybe we're just at this impasse, but evolution is a really dynamic process. And mostly we think that parasites are adapting, hosts are adapting. Where are we in this evolutionary kind of race with these parasites? Is this really a deadlock, or are parasites evolving in response to sickle cell allele? So this talk, I want to go through some of the options and some of the data we're collecting to try to look at this question. So yeah, I can take a poll. How many of you think this is my question? Are parasites evolving to overcome the protective effect of the sickle cell allele? We only have a few. Yes? Yes?
Yeah.
Okay. All right, we're gonna see if you're right. Okay, so I'm gonna give a few, these are the kind of options that I would think could be. So we might say, no, they're not. And that might just be because they cannot. Maybe this is the magic bullet that parasites cannot get around. It's something, it targets something totally crucial to the parasite and they cannot evolve around it. Or maybe the answer is no, and maybe that's because there's no selective pressure for them to evolve around it. Maybe it's not actually bad for them. So it protects people from disease, but maybe it actually isn't bad for the parasite. Another possibility is yes, but this is recent. This is just happening and it hasn't gone to completion yet. And so it's still protective, but parasites are in the process of adapting to it. And then fourth, yes, they are evolving, but something is preventing that that resistance to sickle from spreading. Okay, so I'm going to kind of go through these and see which ones we can cross off. So first, we know that parasites can evolve quickly. So we know this because they evolve very quickly to all anti-malarial drugs that are ever given to them. So this figure is showing different anti-malarial drugs with when they were introduced, and then the line turns lighter yellow when drug resistance was first noted. So you can see over a matter of just a few years, 10 years, sometimes less, there's drug resistance that evolves in parasites. So I'm going to already kind of take this out that it seems unlikely that, though it's possible, that this is a perfect solution and there is no way the parasite could evolve around it. So I'm going to cross this off, at least for now. Okay. One thing I haven't talked about is that actually sickle cell protection is really strong against severe disease. And it's somewhat strong against this uncomplicated mild disease, but it actually has arguably 0% protection against asymptomatic parasitemia, so asymptomatic malaria infection. And so when we look at actually in field settings and places where malaria is both very common and even less common, actually the proportion of malaria infections that are asymptomatic is very high. So this kind of meta-analysis found that between 60 and 99% of infections at a given time are actually asymptomatic. And most of the time, these are just going undetected because most detection is through passive case detection where people come in when they're sick. And so these are just people that are not being affected, but are carrying parasites and can potentially be transmitting them. So this is true across different continents. Most malaria cases, most parasite infections are asymptomatic. So there's also even some evidence that maybe sickle cell is actually better for parasites, that they increase transmission. So this is results from a mosquito infection assay measuring how many mosquitoes are infected when they feed off of blood from sickle cell trait carriers and non-sickle cell carriers. And so I think going in, this was my my guess of what might be going on is that HBS, sickle cell, is a tolerance, confers tolerance rather than resistance. So meaning that these are sort of two different ways that hosts can fight back against pathogens. So they can just kind of reduce pathogen burden. So for a given pathogen load, they reduce the pathogen load. But another strategy is to involve tolerance, where although they have parasites, they limit disease symptoms. And the expectation then is actually quite different for the pathogen. So under resistance, this would put a high selective pressure on pathogen. This is kind of like what drugs do. They reduce the pathogen burden. And so in order to survive, they have to evolve around that. Whereas tolerance actually is not expected to have that same effect. So if the parasite is not causing disease, but is still able to complete its life cycle, then there's no real selective pressure for them to change. And actually some modeling suggests this can actually kind of encourage them or put selective pressure for them to be more virulent. If you have a more tolerant host, you can kind of increase virulence and have more success. Okay, so this is a possibility, and there are even some mechanisms that the mechanisms that propose how this might be happening. So there's still debated how sickle cell is protective from symptoms, but malaria pathogenicity is linked to parasite sequestration and cytoadherence in microvasculature, and it achieves this by, because I'm starting at number 2, but it achieves this by exporting a bunch of proteins to the red blood cell surface, which help the parasite sequester. And so there is reduced expression of those when infections are in AS blood. So the idea is maybe they do less of this, they stay in the circulation more, and they're not adhering and causing all of these downstream consequences that lead to malaria disease. Other possibility is that they don't grow as well. So there is evidence that there is oxygen-dependent growth restriction in AS blood. So the idea being that in low oxygen conditions, maybe there's some degree of hemoglobin polymerization happening, and this is preventing the parasites from replicating as well. And so that just lowers parasitemia and leads to less disease in sickle cell carriers. So then that might think, okay, that's the end of the story. Maybe this is what's happening. It's a tolerance allele. It's preventing symptoms without preventing infection. There's no pressure on the parasite. It really is kind of like an intermediate solution. And unfortunately, it has this consequence in homozygotes of sickle cell disease, and so it can't spread. And so it's more of a problem on the host side. If the host could come up with a solution that didn't have that consequence, it could spread and completely defeat and just become a an asymptomatic infection always. Okay. So mostly though, what people have looked at in trying to understand genetic associations with diseases, host genetics only. So comparing genotypes between cases and controls and looking for variants that are associated with disease or not. But what we hadn't done yet was actually ask the parasites. So I'd say, this is what we decided to do during my postdoc, is ask the parasite, are you doing something to combat the sickle cell allele? You can kind of say that, like, this could be, but without asking the parasite, I think we don't really know. And so one way to ask is to test for association in malaria cases, looking together jointly at host genetic variants, so in this case, sickle cell, and the parasite genome. And so this is what we did. We did a joint genome-wide association study for host and parasite genetic variants in severe malaria cases, sequenced the parasites from 4,440 severe malaria cases, and tested for association between the top, these are the human genetic variants, the top signals from just a host GWAS against the parasite genome. And the result was that we found 3 loci of the 3 parts of the parasite genome that were associated with host genetic variation. So now this is the parasite genome. There are 14 chromosomes. And on the y-axis is evidence of association with any human variant. And it turned out that all 3 of these were associated with a sickle cell allele. So zooming in on these signals, we see that there are association signals with non-synonymous protein coding changes in 3 different regions and 3 different genes. These genes are ACS8 and acetyl-CoA synthetase that is not essential for the parasite, and putative exported protein that is not essential for the parasite, a putative protein phosphatase that is not essential for the parasite. So, oops, did I exit? Okay, it's okay. No. It's my tongue. Just go like this. Sorry. Zoom? No, Zoom is there, but my screen sharing is gone, I think, and it's not. Okay, it's coming back. Sorry. Okay. So it appears that parasites that carry specific alleles at these 3 genes are enriched, are causing disease in people with sickle cell allele, but we don't know almost nothing about what these genes do. They've never been linked to sickle cell before. Okay, so what does this look like? This is what the data actually looks like for this association. Sickle is strongly protective against severe malaria. So these numbers on the bottom are very low. So there are actually only 17 people with severe malaria in the study that carry the sickle cell allele, but they all carry parasites with these particular genotypes at these 3 genes. And in Kenya, there are some more, and they nearly all carry at least one of these alleles at these 3 genes. So another way to look at that is to estimate the protective effect of sickle cell allele against severe malaria, depending on whether parasites carry these alleles or not. And this suggests that another way to a way to summarize this is that parasites that carry none of these alleles, sickle cell is very protective against parasites that carry none of these alleles. It is essentially not protective against parasites that carry all 3 of these alleles. So somehow these parasites seem to be overcoming the protective effect of sickle cell. Yeah.
Falciparum.
This is falciparum.
Are these in Vivax as well?
No.
Valley or
so these alleles are specific. The genes, one of them is specific to like ape infecting malaria parasites. The other 2 are found throughout malaria parasites.
And those alleles are there
in the others? No. So in general, like they don't have the same variation.
Oh,
yeah, it's like they don't even have. So these variants are not even present outside of Africa. That's actually one of the things that suggests they're really tracking. They're pretty much being selected. They look quite clearly like they're being selected for by the presence of the sickle cell allele.
Because most of sickle cell allele, the host, are in Africa.
Yeah.
And maybe Caribbean.
Yeah, there are some others, and so actually it would be very interesting to see if this association extends elsewhere. Yeah, I think we don't know that. Okay, so I'm going to say no to this as well, because if you ask the parasites, I think they are adapting, and so now we have these other 2 possibilities for what might be going on. Maybe this is something recent, and in 20 years' time, sickle will no longer be protective because these alleles will spread throughout the population. Or maybe this is a different kind of deadlock where we are still at some kind of equilibrium. Okay, so after this finding in my lab, we're really interested in asking questions of why parasites are doing this, given that I just explained that there doesn't look like there should be a selective pressure on them because they are still causing asymptomatic infections. And how are they doing this? This ought to be able to give us some insight into what's the key kind of mechanism of protection. If these are getting around it, if we understand how they're doing that, we can understand how sickle cell is protective. And then when is this happening? So that was sort of the first thing we decided to tackle, was to ask whether this is already happening in asymptomatic parasitemia. So why are there all these asymptomatic infections? Some of the reasons are that essentially the reason is that partial immunity develops with exposure. So after people have been exposed a number of times in childhood, they generally can tolerate parasites without getting sick or without getting very sick. And asymptomatic infections are therefore a major reservoir for transmission. They're also more likely to have transmissible parasites, and so they're really a problem for eliminating malaria. So this asymptomatic infection is crucial both for malaria eradication and for understanding this host parasite coevolution, because we really need to look at where the parasite is spending most of its time to understand what the selective pressures are acting on it. And so to do this, we set up a collaboration with a lab at the Center Pasteur in Cameroon, led by Dr. Sandrine Sango. And they already had a really nice study system to look at asymptomatic malaria infections, where they work with schools in this region outside of Yaoundé. And so we worked with them to do a cross-sectional study, collecting samples of children without symptoms from 12 villages over 3 high transmission seasons. We wanted to ask the question of whether HBS, the sickle cell allele, is already associated with parasite genetic variants in asymptomatic infection, essentially asking the question, where are parasites overcoming HBS? Are they overcoming at the step of infection, or is it somewhere later in the disease process? Are these the only ones that can cause disease, or are these the only parasites that can infect people with sickle cell? So this, this is what the study looked like. So we went out to schools, or really the team in camera went out to schools, and included any student who, with parental consent, who was healthy with no fever, and did not report using antimalarials or having malaria symptoms within the past month. Collected biometric data as well as dried blood spot and blood for a microscopy to be able to look for parasites and quantify, have some measure of quantification of both asexual blood stage parasites and these gametocytes, these transmissible parasites. So across the 3 seasons, we collected samples from 2,305 children from the ages of 3 to 17. And the infection rate is about 75% on average. So 75% of children during these high seasons are carrying parasites asymptomatically in their blood, and about 15% are carrying gametocytes, transmissible parasites. And this is in line with what had been seen before. So this is not unusual for this region. Then we took the dried blood spots and extracted DNA, and we developed a protocol to be able to sequence in order to genotype both human genetic variants and parasite genetic variants. So we focus on 4 genes that have been associated with severe malaria, alleles in these 4 genes that have been associated with malaria protection previously. These 2 parasite genes. The 3rd one that I mentioned is actually the variation is only present in East Africa, so it's not in West African parasites. And then we have some others that are they're very polymorphic and are often used to try to estimate how many strains of parasites are present and how related they are. So I'm not going to talk about those so much today, but we have generated this data set for this 2,300 samples. And we first checked that we are kind of getting what we expect, that the number of reads that we get from parasites shows a nice relationship with how many parasites were observed by microscopy. So we do see some parasites in individuals where there were no parasites by microscopy, but that's also common, that there can be low levels that are not detectable by microscopy. So we think this data is looking pretty good. And when we assign, we also can detect individuals that have no parasites. So there are still 20 to 30% of kids who do not have any parasites. And we looked at whether there was a difference in rates of infection between sickle cell genotypes. So this is kind of replicating the report that we see no association between asymptomatic infection and sickle cell genotype. So parasites are infecting people with sickle cell genotype. They are also producing gametocytes at similar rates across all 3 of these seasons that we sampled. The story looks quite different though when we start looking at the parasite genotype. So this is really our major result, which is that when we type these alleles, so that the rates of infection are the same, but the parasites are almost completely different. So this is actually what we see at one of the loci that was found in that it seems to overcome sickle cell protection. We found that in these, I don't think I have the numbers here, there are about 450 sickle cell trait carriers that have asymptomatic infection, and 78% of them carry parasites with this allele, this V7 allele at phosphatase. And in the 1,500 AA individuals who are infected, only 5% of them carry that allele. So this is just extremely significant. And I think very remarkable that even though the rates of infection are the same, they are different parasites that are infecting sickle cell and non-sickle
Did
cell
you split
carriers.
that by village, or, there's all these individual locations?
Yeah, we should do that. We haven't done that.
Because maybe it's more about the family and the concentrations of the parasite that individual, I mean, this is just
Yeah.
the effect that probably isn't, but
Yeah, we have done some things to try to look at whether there's sort of structure in the parasite data that might explain it, like that you have like correlated population structure or something. But just with it doesn't look like it. For the samples we have RNA-seq, we have kind of genome-wide data that we've tried to do that for. So I don't, it's quite a small geographic range that it's hard to imagine, but that's something we should look at more. But we can split it by season. We can look at kind of whether there's any other metadata that explain this, and I don't think so. We also have other human genetic variants, so we can look like, is ABO related, and we see nothing. It really seems to be specific to HBS. And so we have our two loci. So if we look at both of them, so this is looking at them separately, we see the same effect, perhaps a little bit less strong at this variant in ACS8. And we see that these are in linkage disequilibrium with each other, so they tend to be found together. It's even, if you combine them together, you tend to see both of these alleles more often than expected, either both of them together in AS or both of them, or neither of them in AA genotype. Okay. So this kind of is making us rethink this coevolutionary model. So actually now we think, we're kind of thinking about this as instead that maybe when sickle cell arose, it did confer resistance against all parasites. Parasites could not infect. And that did put a selective pressure on the parasites, and they came up with this solution. And now we see this solution. But then if that's the case, then we have the question of why this parasite alleles don't just become fixed. If they're able to infect both AA and AS, and the others cannot infect AS, these should spread through the population and fix. But instead, what we see is a correlation of the frequency where parasite alleles track sickle cell allele frequency. So that suggests that, I'm going through this first. Essentially, this suggests there may be some cost. There may be some cost to these alleles, and that's why they cannot fix. So that's what I'm going to try to take you through now. But there's some additional evidence that these may not be recent alleles that are spreading. So there are, if this was a new allele that was spreading through the population, we might expect to see some kind of population genetic signatures around it that that are expected if you have strong selection. So that the idea is that when a new advantageous allele appears and it spreads through the population, it drags with it genetic variants nearby. And so people have done these kind of scans to look for that in parasites, and we can see that these do not show that kind of signal. You can almost see that in the association plots where there's a very narrow peak. It's not a huge set of variants that are all being carried together. However, it does show some other unusual signatures, and one of them is frequency differentiation. So if we look at the allele frequency across populations, so these alleles are actually not found outside of Africa. So this has come up previously in kind of scans looking for variants in the whole genome that show extreme differences in frequency across populations. And it actually comes up both when comparing Africa and Asia, and also within Africa. So this variation within Africa is also more than you see at other places in the genome.
You don't have the Americans on there.
Yeah.
There are African descendants in the Americas.
And there are, some of these are present there.
These
alleles are present there, yeah. So
you just don't, okay.
Yeah, they're just not, they weren't in this data set. So they are in some more recent data sets. So it's something that we have looked at a little bit because they show a somewhat different pattern. And then lastly, we in our lab have also tried some other tests kind of focusing on this idea that maybe this is a long-standing kind of, these have been polymorphic for many years. And so one signature that we expected these have been polymorphic for a long time is kind of a buildup of alleles at a similar frequency nearby. So there's a test we applied to look for this, and we find actually that ACS8 shows a signature of this that stands out from the rest of the genome. So we think this suggests that it's more likely that this is not new, that this has been ongoing for thousands of years potentially. And then the question becomes, well, why isn't it spreading? What is preventing that? And then how might that change as the picture of malaria changes in the future? So I think our working model is that there is some cost involved to these parasites. And so maybe parasites, they can't predict whether they're going to be in an AA or an AS host. So sometimes they find themselves in one versus the other, and one allele may have an advantage in an AA host and one may have an advantage in an AS host. And so actually they're at some equilibrium frequency such that the infection rate is the same. So I think actually this might be a perfect kind of balance, and that's what you'd expect. That's something we're working on, trying to model what parameters would lead to that kind of equilibrium where you would have the same infection frequency overall. Okay, so other things we've been trying to do in the lab is to look then functionally at how these infections differ and whether we can detect some evidence for cost. And so we've done this through, we're doing some looking at gene expression, and we're also trying to see if there's an effect on transmission success. And so for 63 of the individuals who participated in the study I just talked about, we also collected venous blood, and we're able to do both of these experiments. So both bulk RNA sequencing to look at gene expression, where we can look at both parasite gene expression and host immune cell gene expression, and then direct membrane feeding assays, which were carried out by the lab in Cameroon, where they feed the infected donor blood to mosquitoes in a cup, wait for a week, and then dissect the mosquitoes to count how many mosquitoes were infected and how many oocysts they could find. Okay, so I'm going to just briefly summarize where we're at now and what our potential findings are. So we can look at whether there's a difference in the kind of infectivity of when mosquitoes are fed on blood from AA or AS hosts. So this is similar to the result I showed at the beginning that had been reported that AS carriers might be better transmitters. We don't see evidence for them being better transmitters here. Instead, what we see suggestion of, I guess that potentially this allele may have a cost to transmission, that these are less good transmitters. Of course, these are very correlated with each other. So you can sort of see they're similar, but we have a few cases where an AS individual is not carrying the allele, and those are exactly the ones that are having the opposite effect on transmission. Okay. And then secondly, we were looking at gene expression, doing this dual transcriptomics, where we have bulk RNA sequencing. We capture together both parasite and human RNA. And then we computationally separate those. We can use computational techniques to try to figure out what proportion of cells were present. And so we can do that both for human immune cells and for different parasite stages. So it's proof of concept that we can do this. So this one first. In parasites, we can see that the proportion of gametocytes we infer, these are ordered from high gametocytemia by microscopy on the left to low, that we infer higher gametocyte proportions in this yellow and red color in samples that we also saw more gametocytes in microscopy. So we think this is working pretty well. We see not much difference across individuals in immune response. And then we can look for whether there are differentially expressed genes in both human and parasite between different genotypes. So mainly what we see is that there are a lot of genes that are differentially expressed between sickle cell trait and non-sickle cell trait people. That in itself is kind of interesting because there actually hasn't been too much work done on sickle cell trait, sort of generally thought to be asymptomatic, but actually we see there are quite a lot of genes that are differentially expressed. In the parasite, And between parasite genotypes, we see quite a lot. We don't see very much. So I think maybe this isn't too much by transcriptional response, with the exception of the phosphatase, which actually the genotype is associated with increased expression. So it does seem that there is increased expression of this phosphatase when parasites carry this other allele. And then lastly, I just want to mention that a whole other area that we're exploring is to try to functionally characterize differences between parasites in the lab when grown in AA and AS or SS donor blood. And so this is something we're doing together with Paul Sickle's lab in biochemistry here. We have a joint postdoc who's working on this, and we have a collaborator at Washington University that works with blood donors. And we do see that there is evidence that parasites, so FUP parasites, carry these alleles. They can grow in SS blood at low oxygen, where wild type parasites cannot. So there is some evidence that they are overcoming this low oxygen growth effect of sickle cell allele on parasites. Okay, so that's all. We're very much in process of understanding this, but we think it is a kind of major player in what's going on in this interaction, that we think these sickle cell and parasite alleles may be at a coevolutionary equilibrium, that parasites can overcome HPS protection and that we see them already highly enriched in asymptomatic infections. Yeah, so I guess I'll acknowledge everybody in my lab, especially, Helena Hobson, who's a PhD student who's led this project, and she's shown here with the lab in Cameroon, who also have done lots of great work collecting this data and working with us on this project. Thank you.
Thanks, Ellen.
Yeah.
Yeah.
Any other questions?
Origin story.
The people that get sick, not the asymptomatic, how long do they have the infection in them, do you know?
So it varies, I guess. You have an answer for this? I mean, like, how long does it take them to become symptomatic and then develop disease?
Well, yeah, so they're not, if they get bit by a mosquito, I assume the gamete or whatever is not there the rest of their life.
No, they usually clear them. So it's sort of how long they can, so it can become an asymptomatic infection that becomes cleared. It can become an asymptomatic infection that turns into symptomatic infection. I guess those are kind of the options without any drug intervention.
Well, you can treat it with a drug and it will go away within a few days.
Yeah. Yeah.
So the asymptomatic and the symptomatic are infectious for the same length of time.
That's a good question. Yeah.
Yeah, I think that's something we don't know for sure. There's sort of the idea that maybe symptoms are associated with more infectivity, because that could explain why, otherwise why bother having any symptoms? So like the more parasites, the more they should be infectious, and usually more parasites leads to disease. And so there may be some relationship between symptoms and being more infectious, but I think we don't really know that.
Catered for the rest of the world.
Yeah. Yeah, I mean, a lot of it's by mosquito control. I think you get rid of the vector has been the most successful way to get rid of malaria.
Draining the swamps in the southern US was how they got rid of it. And
yeah.
all the way from Philadelphia down to Louisiana, to getting rid of mosquitoes, and then I think from what I hear, places who've really done well, it's deep, spraying insecticide using bed nets and just eliminating it. Because like Sri Lanka became eradicated in 2015 or 2020 or something like that. Yeah. So, because it's an island, and so they were able to get rid of all the mosquito that bear malaria. And so
Yeah.
they eradicated it, but it's probably not the best example because it's an island and you can,
Yeah, you essentially have to get rid of all the mosquitoes and all the infections. I mean, people try, there's Mass Drug Administration tries to just at the beginning of the season, treat everybody and so lower the parasite load. But I think this is one of the issues that people don't, if you don't treat everybody, there's still, I know there's a,
oh,
sorry, we have it at 1.
ah,
Yeah.